There we go. Most of you are wearing an article of clothing that has a color other than, say, white or beige or brown or others. And that is a product of the textile industry, and the textile industry is very good at making cloth that's colorful. We take it for granted, right? You go to, if any of you were unfortunate enough to go to Hobby Lobby in the fabric section, you'll see all kinds of fun designs on fabrics, and you can make Buzz Lightyear jammies for your toddler and all of this stuff enabled by the fact that we have colorful molecules. And what makes a molecule colorful is its extent of conjugation. You probably learned in 351 that the more highly conjugated a molecule is, the narrower the gap between HOMO and LUMO, and that eventually gets to a point where photons of light in the visible region, that have wavelengths in the visible region, are sufficient to promote an electron from HOMO to LUMO. That molecule then absorbs that wavelength of light, and then it reflects other wavelengths of light, and you see color, right? The molecule and things that are dyed with that molecule appear colorful. In thousands of years ago, the available things for coloring cloth were rather limited, especially if you wanted deep and dark colors, like a deep purple or red. You can get some of these from natural sources, but anytime you're isolating a molecule from natural sources, probably you're grinding up like gazillions of flower petals and so on, and it's quite tedious. So here's an example of indigo. It's a highly conjugated molecule. It's blue. You can get it from the plant that's aptly named indigo. But, imagine how much dye you're going to get out of each one of those petals, and you have an idea of how much you have to grow and harvest to even get enough dye to make something blue. This purple dye, Tyrion purple, is actually biblical. It's not that different from indigo. It just has a bromo group at these positions instead of a hydrogen. This was isolated from the shell of a Mediterranean sea snail. And I say that it's biblical because this dye is mentioned in Acts. When Paul and Silas visit Philippi, they meet a woman named Lydia, who's Jewish and married to a Greek, and she's called a seller of purple, which means, I don't know if she actually made the dye herself or was just trafficking in the dye and selling it. But in any case, somebody was diving to the bottom of the Mediterranean, well, probably not the very bottom. Anyway, somebody was diving to recover these sea snails. They were crushing the shells, grinding them up, and extracting the color, extracting the dye. And this was tedious and expensive, which is why purple is a color that's associated with kingship, emperors, because it was just so expensive to get. And then here's another dye that's bright red that you can get. I don't actually even know what the madder plant is. So there you are. And I suppose, anyway, so dye molecules were precious and difficult to get. And this was the way things were until the mid-1800s. So at that point, a young 18-year-old British kid, William Henry Perkin, was working with an advisor in chemistry projects. And like most 18-year-olds, he probably overestimated his competence. So he knew that there was a molecule called quinine that was isolated from natural sources and which could be used as an anti-malarial molecule. There's a lot of things we take for granted today living in America, living in the United States, and especially living in the Intermountain West where things are dry. But malaria is a scourge of the earth, and tons of people die from it, and it was way worse back before we had anti-malarial drugs. And so the idea of taking a molecule that was useful, but figuring out a way to synthesize it so you didn't have to isolate it from its natural source was attractive. At the time, though, chemists didn't have the tools we have today. So they could determine pretty well the relative amount of carbon, nitrogen, oxygen, and hydrogen in a given molecule. But connectivity was another issue entirely. And Perkin had this idea. See, he noticed that the molecular formula of quinine, which I haven't drawn here because it's not important to our discussion, had the same number and kinds of atoms as if you had taken 3 molecules of aniline and put them together in one molecule. So he had this great idea. What if I take aniline and I mix it together with some oxidizing agent? Maybe I'll get quinine out of it. Because why not, right? Like, what could possibly go wrong? Well, it turns out the aniline sample he was using was contaminated with these other related compounds. And you can imagine that these would be tough to separate. They differ only in the addition of a methyl group at the ortho or para positions. Don't worry about the names of these. I will use them, but mostly just ortho and para methyl aniline. You can imagine it'd be difficult to separate these because they're very similar in polarity and so on. Probably difficult to distill one from another. So Perkin started to work, and he put his sample of aniline in a flask with potassium dichromate and sulfuric acid. Okay? And you have seen this reagent before in 351, where you were told you can use it to oxidize primary alcohols to carboxylic acids, and you can also use it to oxidize secondary alcohols to ketones. I am not sure why he decided to try it, why he thought he needed an oxidizing agent, because he had no idea what the connectivity of the molecule in quinine was. But in any case, he tried the reaction, and as with most things in organic chemistry where you use sulfuric acid, what crashed out of the reaction mixture was not quinine, but rather this chunky black solid. And usually when that happens, when it looks like you've got like charcoal at the bottom of your flask, you throw your hands up and say, dang it, I guess that didn't work, and you clean it out and try something else. And usually it's difficult to clean out a flask that has that kind of gooey, tarry, chunky black solid in it because it may not be that soluble in either water or organic solvent, so you sort of have to mechanically chunk at it, and it's a disaster. And actually, if you're in a well-funded lab and your advisor isn't looking, you actually just throw the flask, you actually just break the flask, and then you don't have to clean it. So I'm surprised that 18-year-old didn't do that. He actually said, okay, I got to clean this now. He started rinsing out that black solid with alcohol, probably attempting to find a solvent that would dissolve it. And he noticed instantly the solvent, the alcohol he put in there to rinse the black solid was purple. And instead of thinking, oh, gee, that's kind of cool, or that's weird, he thought, opportunity. Because at that time, the only way to get purple dye was from natural sources, and he had just found a way to get it from chemicals in a simple process. And so he named the molecule Mauvine, after the color mauve. I'm not actually even sure how to say that color. And he found that it was able to dye silks and textiles. He patented it. In 1857, at the age of 19, he opened a factory in England to make this stuff. And he retired a wealthy man at age 36. So this is kind of a crazy serendipity study. He devoted the rest of his life to chemical research, and there's still to this day a prize, one of the most prestigious prizes awarded by the American Chemical Society, is named after this guy. So serendipity is a real thing, and It's not your average 18-year-old that with bravado and confidence says, sure, I'm going to do that, and then discovers a molecule that like changes the whole world. Yeah?
Did he do anything else of note in his life, or was that?
Did he do anything else of note in his life, or was that it? You know, if you stumble upon a discovery and make $1 gazillion, then that's what you're known for. And no, actually, there is a history after that. I just don't know it that well.
I mean, I'd be okay with that.
Yeah. Yeah. Yeah, there is a history after that. He did chemical research for the rest of his life. The Royal Society of Chemistry in Britain, for a while, had a couple of journals, academic journals, named after him. Sort of the father, and sort of the father of modern chemical research, because when you discover something cool, everybody else looks at that and says, ah, dang, I could have done that. Probably there were a few other people that said, shoot, I threw away a flask with black chunky solid in the bottom of it after trying just the same thing. And so being jealous that you didn't discover it and it now seems obvious in retrospect, people try to make improvements, and that's where innovation starts to happen. So the discovery of Mauvine, this purple dye, led to a surge of research in all areas of organic chemistry, other types of dyes, perfumes, anesthetics, and also drug molecules. And the relationship between dyes and drug molecules is not obvious, but it's actually really strong and really important. But before we do that, let me just show you a picture of an original sample of Mauvine. It's labeled as original, which means that's what it was when he put it in there. I don't know its stability over long periods of time, but it's still purple. Prepared by William Perkin in 1856. And there's the cork, and here is a fabric that was dyed with Mauvine. So you can see it's not just it's a pretty deep purple, the kind that Caesar would have been envious of, right? And now it's available to people across the world, even without Caesar's wealth and opportunities. So here is the structure of Mauvine that was discovered. Again, this is also a tradition in chemistry. We often discover that a molecule is useful, and we often use it before we know what its structure is and before we know how it works. Thankfully, the FDA has sort of crimped down on that now. We have to know the structure of a molecule. We have to know what it does. We have to demonstrate that it's safe. But anyway, this dye was used long before anybody knew what it was. But and it's actually a mixture of compounds. This is just one of the Mauvine molecules. But if you look at its structure, you can see portions of it that come from these precursors that Perkin was mixing together under those oxidizing and acidic conditions. So here's in orange aniline, a 6-membered benzene ring with one nitrogen attached to it. You can see that structure here and here. So it looks like 2 molecules of aniline ended up in Mauvine. In purple, we have the ortho-methyl aniline, and you can see that substructure here. And then in blue, that's not blue, sorry, that's green. Sorry, I just got distracted by the annoying philosophical question as to whether the color I perceive as green is the same color that somebody else perceives as green. Anyway, who knows? But this is science, not philosophy. So you can see this structure in green comes from the para-methyl aniline. And in red are the bonds we had to have made. And now you can sort of see where some of the oxidation chemistry needed to come from. For example, this bond between carbon and nitrogen used to be a carbon-hydrogen bond. And in chemistry, oxidation often consists of reducing the number of carbon-hydrogen bonds and increasing the number of carbon heteroatom bonds. So you needed some sort of oxidizing agent to make these new carbon-nitrogen bonds. I'm going to show you one such reaction. The first part is going to be the preparation of the electrophile. That part you can basically ignore. The second part is going to be an electrophilic aromatic substitution reaction that makes the new bond. So to begin with, we'll start with the para-methyl aniline. Sometimes oxidation chemistry happens in mysterious ways. So some metals are so desperate, as it were, for electrons that they can actually do what's called single electron transfer, where an electron literally hops from the molecule onto the metal. That doesn't happen a lot, and we're not going to do it hardly ever in this class, but that's likely what happened here. To generate an electron-poor molecule that was a radical and a cation, and then in another step of oxidation becomes this sort of imine, this weird imine. It's highly conjugated, and it's electrophilic at this nitrogen. That's exactly backwards that you would expect from an imine. You'd think it would attack the carbonyl carbon. That's why I'm saying don't worry about this. But a good nucleophile could attack here at the nitrogen. That would push these electrons here and here. That restores aromaticity, so that's sort of a driving force for this reaction. And then presumably you could pick up a proton from your acid at that stage. So with the idea that, what the heck does that stand for? MRA. I got nothing. With the idea that this molecule is electrophilic, then now we're going to use the orange aniline as a nucleophile in an electrophilic aromatic substitution. So pi electrons from benzene attack the electrophile. These arrows are just how that electrophile reacts. You don't need to worry about them, but this one is the electrophilic aromatic substitution. We get that familiar intermediate where we have both the electrophile and a hydrogen on the same carbon with an adjacent positive charge. And when we remove that proton with a base, probably the conjugate base of our acid, we restore aromaticity. And this happens a couple of other times to make all of the carbon nitrogen bonds in this molecule. So though he didn't know it at the time, Perkin was using a class of reactions that we've studied in chapter 20. But as I said, whenever a new discovery is made, people want to find newer and better ways of doing what that molecule does. That's how chemical research and the chemical industry functions. This is how you argue to the National Institutes of Health that they should give you money to fund your research as a professor. You tell them, hey, what's currently done is this, and that's good, but we have a better way. And you try to sell them on how much better your way is, and it's sometimes works and sometimes it doesn't. But this leads to incremental improvements and sometimes transformative improvements in technology. So enter a class of molecules that are called Azo compounds. I don't have any details for you on the discovery of these molecules, only that people were working with similar compounds and discovered that they too were highly colorful. So here are some examples of some Azo dyes that have bright and intense colors. Here is one called Para Red. In an Azo compound, you have two nitrogens linked together via a sigma bond and a pi bond, and then each nitrogen is itself bonded to usually an aromatic ring. Usually with alkyl groups, these compounds aren't stable for reasons we'll talk about later. And you can begin to see how somebody might have come across this. Maybe they made the phenyl azo compound and then said, I wonder what would happen. I wonder if the color would change if I installed a nitro group on one of those benzene rings. I'll show you later how to make, that's the new chemistry. We're going to talk about how to make the azo compounds. But you know how to put a nitro group on a benzene ring. It's pretty easy. And so somebody could have said, hey, let's just try it and see what happens. And you can also see that they may have thought, hey, instead of a benzene ring here, why not try naphthalene? Let's see what that would do. And this leads to a very important idea in chemistry and biology, the idea that molecular structure is tied to molecular function. And that seems simple, like duh, what a molecule looks like, what its shape is, what its stability is, should influence its function. But the idea that comes from that is if I modify the structure, I have the chance to change the function. And so what we've been learning in OChem is all these systematic ways to modify the structure of molecules with presumably the goal of changing their function and discovering new and useful functions. This kind of thing is sometimes called structure-activity relationships, and to this day, medicinal chemists in the pharmaceutical industry will take a lead compound that they think has some interesting activity and say, what if we change that hydrogen to a fluorine? Or, what if we put an ethyl group there instead of a methyl group? They'll try a bunch of things, they'll screen lots of compounds, and they'll see what works. And this idea was started in the days of the dye industry. So here's Congo Red. It is even more elaborated now. It's got 2 azo groups in it, two benzene rings attached to each other, attached to azo groups, and then these other arenes on the side. But look at the functional groups there. NH2, you know how to put that on an aromatic ring, right? You install the nitro group and then you do reduction to get the aniline. You also know how to put this sulfonic acid group on a ring. That's the sulfonation reaction we learned. And so through these tools, people were able to test, let's see if we add this group, let's see what happens. By the way, sometimes people are critical of let's see what happens science. They call it, especially grant reviewers, they call this a fishing expedition where we're just going to try a bunch of things and see what happens. It's not very inspiring to somebody who's wanting to see whether they should invest money in your research. But in a lot of cases, that's how the actual scientific discoveries are made, is let's just try a few things and see what happens. It's always better if you have a hypothesis, but often the initial discovery is, whoa, we tried something and something happened. So about this time, in the late 1800s, maybe the 1870s, German chemist and physician Paul Ehrlich was working on a project to use dye molecules to stain bacteria and tissue samples to make them more visible for microscopy. If you've ever looked at a sample of cells under a microscope without any stain, they look kind of boring because they're transparent, they're sort of colorless, and you can't see any of the intracellular organelles very well. But in some cases, if you add a dye and stain the cells with a particular dye, that dye molecule will be taken up more strongly in some membranes than in the cytosol, and so then you can start to see the membrane of the cell and the intracellular organelles like the mitochondria or the nuclei. People still do this today. It's called histochemistry, and the techniques are really advanced now, and you can use fluorescent molecules and confocal microscopy. But back in the day, he was just looking for stains for bacteria and tissue samples. So he happened upon this molecule methylene blue, which is shown here. And this in the 9 o'clock class, when I saw that structure of methylene blue and I heard the name, I suddenly remembered something that happened my freshman year, which I'm now hesitant to share with you based on the reaction that I got from the 9 o'clock class. But I guess I will not learn from my mistakes. I will throw caution to the wind. Methylene blue, so it's a blue dye, and though it has some side effects, you can ingest it. In fact, we're going to later talk about how Paul Ehrlich discovered that this methylene blue can actually kill the Plasmodium parasite that causes malaria. But, so at about this time of year, I think in my freshman ward, I lived in Deseret Towers, which is no longer there. And there was this guy and this girl that really liked each other, and they showed this by pranking each other because we were apparently in kindergarten or something. And I can't remember, I think it was she that played the prank on him. She delivered a plate of brownies to his dorm, but the brownies were laced with or soaked with methylene blue. Which it must not have been dripping because I don't think he noticed. Or if he noticed, he just did it anyway. And of course, it was supposed to surprise him because the methylene blue gets filtered out through the kidneys fairly quickly, and then suddenly you're in the restroom and discovering something really surprising, which is that your urine is blue. So then somebody in the audience asks me, so how dangerous is methylene blue for people? And I got the sense that he was actually having an idea of how he might use methylene blue. So I said, extremely dangerous, do not do it. Please do not administer chemicals to people without their knowledge. That's horrible. I was not involved in this. At worst, I'm an accessory after the fact. And now you will all virtually sign a waiver that agrees to indemnify and hold me blameless in perpetuity for any effects of bad decisions you make with methylene blue or chemicals of any kind. All right, go ahead.
So, asking for a friend. Let's say someone, signed a waiver saying that they wouldn't mind if they took it.
Asking for a friend. What if you took it? There are side effects. So with anything, you should check side effects before you ingest something. You should also check and see whether it's FDA approved. Because, go ahead. Did he get the girl? No. Well, they continued to date until the end of their freshman year, whereupon he went on a mission and they grew apart, and that was that. I will say that was the year I met Amber. She did not lace brownies with methylene blue. She and I continued to write during the mission, and we are now married for 20 years. So you can see the consequences of... No, has nothing to do with it. By the way, I'm not a fan of waiting for somebody on the mission. So she was pursuing her own stuff, and then... Oh, dang. I guess I'm going to have to tell another story. I apologize. Yeah, go ahead.
So, my process of flawed logic. If I convince my friend to lace some brownies with methylene blue and give them to their significant other, the chance of me finding the person to marry will increase.
I think that's what we concluded from that discussion. Yeah, go ahead.
Just so everyone knows, according to the Mayo Clinic about methylene blue, there's like dark urine in addition to diarrhea, fever, headache, dizziness, pale skin, poor coordination, rapid heart rate, redness of skin, sweating, short throat, shivering, tightness in the chest, trembling, shaking, fatigue, etc., etc. And then there's another long list for symptoms of overdose.
Okay, so sounds like you want to avoid it. I'm sorry, I am going to tell another story now. I know you don't mind because you don't really want to learn chemistry today. So Amber did date a guy while I was on the mission, and boo, right? She'd send me pictures. I mean, she didn't actually think it was dating. Amber grew up with brothers and never really got the idea that some guys don't know how to just be friends. So he was definitely interested in her. She wasn't necessarily interested in him. And he got it in his mind that the only reason she didn't like him was because she was still holding out hope for me. So I kid you not, the kid writes me in the mission field. He quick writes down my address from one of the letters that I sent Amber. And he writes me this very long letter detailing how he loves her and he knows he's right for her, but the only thing holding her back is me. And so I need to look deep in my heart and release her from my clutches so that he can be happy ever after.
Where were you serving?
I was serving in McAllen, Texas at the time. Oh, really? How old's your dad?
You served like mid-90s.
Okay, I served late 90s, so 98 to 2000. So. Okay, we would have missed each other, but he may have known my mission president. Anyway, so I'm serving in Far, Texas, and I get this letter, and I read it, and I'm like, that's kind of funny. That's kind of weird, because Amber and I had never agreed that we were waiting. We didn't say we loved each other. We were keeping everything, just like we were supposed to in the missionary guide and whatnot. So she finds out that he's written a letter, and she's totally ticked off. So she writes me a letter saying, so I'm really kind of embarrassed that he did that. By the way, we've never really talked about it. I mean, I do like you, but I'm not waiting for you. But when you get home, we'll see how things work out. But that was later. I'm skipping ahead to that was months later. The day I got this letter happened to be interviews with the mission president, and he would take time with us one-on-one, and you could sit and talk to him however long you wanted. And so, at the end of this interview, he says, "Is there anything else?" And I say, "Well, I don't want to get all soap opera on you, but," and I told him the story of this letter. And I thought he was going to give me something about lock your heart, elder, and there's just nothing. Instead, he sort of smiled and he sat back and he says, and he was an attorney, actually. And so he said, you should do nothing. Because I was like, should I write back to him and tell him? And he says, you should do nothing. Because if you write back to him and tell him that, well, in actuality, you and Amber have no particular arrangement, he's going to use that against you. So just pretend like you never had the letter. And it turns out that was the right thing to do because, again, weeks later, Amber found out that he'd written the letter and she was ticked off at him. And the rest, as they say, is history. So that story took way shorter than I thought it would. That's really good. So I'm sorry about that. I do have a tendency to go off, but whatever. So back to methylene blue. Paul Ehrlich he has this idea, he discovers it stains the Plasmodium parasite that causes malaria. And he thinks if this molecule stains the parasite, that means it can get in the parasite. If it can get in the parasite, it might be able to disrupt the processes that keep the parasite alive. Presumably, when he was staining the samples, he wasn't working with live parasite, probably because they were dead after he stained them. I don't know. But he tried it, and it worked. And again, this was in a day where you didn't have to have informed consent and get IRB approval to try things on people. But they gave methylene blue to people who had malaria, and they got better. And so this was the birth of the idea that a specific molecule could be used to treat a specific disease. This was the beginning of the magic bullet idea that you could have a molecule that would go in there and attack a particular pathogen or stop a particular cellular process without doing other stuff, without having undesired side effects. So with that idea in mind, and the idea of structure and function relationships, it was just a matter of time. Let's see how we can alter the structure of dyes to see if they can kill things that they were previously staining. So in the early 1900s, Ehrlich and others developed this molecule Trypan Red, which is a red dye. And again, you can sort of see the evolution of structure if you compare this to Para Red and Congo Red. We have the same biphenyl core, the same diazos. We have the same naphthyl groups. We're putting other sulfonyl groups on other portions. That killed trypanosome parasites, which I think is sleeping sickness. I'm not sure, but anyway, very important. Later, they discovered this molecule arsphenamine, which is an arsenic derivative of an azo compound. So I don't often think about arsenic, but when I do, I notice that it's on the same column of the periodic table as nitrogen. So you can get the idea that this might be a similar functional group to having the two nitrogens double bonded to each other. Turns out the structure of this molecule, it turns out to be a little bit more complicated than this, but they demonstrated that this molecule could kill the bacteria that causes syphilis. And syphilis is a terrible disease that you get, and it leads to all kinds of degeneration, and but now could be cured. That very quickly became the most widely prescribed drug in the world, which, parenthetically, if I might editorialize, suggests that the Victorian sensibilities about sexuality were not necessarily reflected in the behavior of people back then, if syphilis was so widely needed to be treated. Anyway, so there you go. That's the story, and sort of modern chemotherapy grows out of this. If you want a good read on cancer and how what I've told you today relates to cancer treatment, you could look at the book The Emperor of All Maladies by Sid Mukherjee, and he takes you through the history of cancer discovered in mummies from thousands of years ago up through the present. It's pretty interesting. Now, in the last little bit, we're going to do a little bit of chemistry. We're going to talk about how to make Azo compounds. And it starts where we ended in chapter 20. That is, we know how to put a nitro group on a ring, and we know how to reduce that nitro group to an amine with hydrogen and palladium on carbon. But how do we get from there to something where we have that nitrogen also bonded to another nitrogen and via a double bond, not just a single bond? The process of doing this begins with something called diazotization. You don't need to know that word. I just mention it because I learned about this in 352 when I took it a gazillion years ago, and I promptly forgot it. And it came back to bite me on a test, and so I've always kicked myself over that, and so now I want to make sure I understand it really good because it matters to me for some reason. I don't know. So this process is kind of interesting. You use the aniline, and then you use a form of nitrous acid. You start with sodium nitrite, and then you use HCl in aqueous conditions. Now, nitrite looks like this. Nitrous acid is a weak acid, and it's also highly unstable. HCl protonates this oxygen to become the OH group, and that's nitrous acid, which some people call HONO. But the HCl can also protonate that oxygen again, which turns the OH group into now a good leaving group. And you've got lone pairs on the atom adjacent to the nitrogen that bears the leaving group, one of the lone pairs on oxygen. These can actually do an elimination reaction where they kick down and kick off the water leaving group, and that generates the following cation, which is called the Nitrosonium Ion. You don't need to know that name. It's electrophilic. Oops, that is a wrong Lewis structure. I put the positive charge in the wrong place. It's electrophilic on nitrogen. You can draw a resonance structure in which the positive charge is on the nitrogen instead of on the oxygen. And of course, nitrogen is less electronegative than oxygen. Okay, so what is a nucleophile going to do? Well, if you put the aniline there, the nitrogen lone pair, reactive lone pair, is going to attack the electron poor nitrogen of the nitrosonium ion, and that, is how we make our first nitrogen-nitrogen bond. Now, in the interests of efficiency, I will say, yes, you have this intermediate and you remove a proton, probably with the conjugate base of our acid, and you get something that's called a nitrosamine. Your book talks about these a little bit because some of them are carcinogenic, and if you've ever been warned about eating hot dogs at some point, it used to be, I think during Amber's pregnancies, it used to be the doctors would say, hey, don't really eat hot dogs, and the justification for that was they have nitrites in them, and those nitrites can presumably form nitrosamine compounds, some of which are carcinogenic. So you may have heard of nitrites before, and what they're doing is what I'm showing you here. But then what happens, because remember we're under acidic conditions, is likely, and this mechanism I'm only showing you as background, you don't need to know the mechanism. Likely we protonate that oxygen, and then a base comes and removes this proton, and we make a new nitrogen-nitrogen pi bond, and then likely that process happens again, only this time we protonate the OH group to turn it into a good leaving group, and then lone pairs on the nitrogen kick down, water leaves, and we're left with the product, which looks like this. And this is called, and the counter ion is Cl-, this is called a diazonium salt. Okay. So this is a precursor to those azo compounds that I told you about. It's a new kind of functional group to install on a benzene ring. One of the interesting things about it is that you've basically got nitrogen as a leaving group. So you might expect this molecule to be highly electrophilic. And it is. Someone in the first hour asked, what are the regioselective preferences of this N2 plus group? I said you'd expect it to be a resonance electron withdrawing group, so presumably a meta-director. However, I think it's reactive enough that if we want to do this, we actually would put other groups on here first and then make the diazonium salt. So I don't think you're going to have to deal with regioselective preferences of either the diazonium salt or the azo compound. But let me just show you briefly what you can do with this electrophile. You remember that I think somebody asked earlier, do we know how to make phenol? That is to install an OH group. Remember, to make the aniline, we had to first install the nitro and then reduce it. Do we have anything that does this for oxygen, for the OH group? Well, if we start with the diazonium salt, we can do a reaction that's called nucleophilic aromatic substitution. This carbon is electron poor. Oxygen can attack it. Now, remember that you don't ever really do SN2 reactions at sp2 hybridized carbons, in part because SN2 reactions proceed from backside attack, and backside would be inside the benzene ring and the OH can't get there. So this mechanism is really sort of carbonyl-like. It's an addition followed by elimination reaction. We're going to have OH attack that carbon, and we're going to break one of the pi bonds of the ring. Effectively, what we're doing there is OH is dumping its electron density into one of the empty pi orbitals of the benzene pi system. That temporarily breaks aromaticity and puts a negative charge on this carbon, which is stabilized by resonance. I'm not going to draw those resonance structures, but hopefully you can see how we could delocalize that negative charge at these carbons. At this stage, though, we now have, as I said, a great leaving group in the form of nitrogen, N2, and an adjacent lone pair that can kick down and do the elimination reaction, releasing nitrogen, which bubbles off as a gas, and giving you phenol. So this is our first way of actually making phenol, which is kind of cool. But you can do a lot of other things, which we'll talk more about, I think, next time. But if you use copper iodide you can do a similar reaction, substitution reaction, in which you put an iodo group on the ring. We did not have a method. We had a chlorination and a bromination method. We did not have an iodination method because it doesn't work for some reason for iodine. You can also do this with fluorine. And again, we did not have a fluorination method like we did for chlorination and bromination, in part because F2 is just so violently reactive. The reagent you use here, we'll talk more about later. It's HBF4. I don't know how it works, and the text doesn't give you any mechanism. But that is, if you wanted to install fluoro groups on a ring, that's one way you can do it. Okay, so that's not Azo compounds. Let's show you how you can make an Azo compound from a diazonium salt. Any questions you want to ask at this stage? And we'll come back to these reactions again next time as we practice some synthesis. So we've said that the diazonium salt is an electrophilic compound. Got the positive formal charge on that nitrogen. So you might expect this terminal nitrogen to be electrophilic, because if a nucleophile attacked here, then we could move these electrons from a pi bond onto the nitrogen as a lone pair, then we'd have something that was neutral. So we're going to do this with an aromatic ring. You can actually do, and so what I'm going to show you turns out to be an electrophilic aromatic substitution reaction, only we're using the diazonium salt as an electrophile. So pi electrons from the benzene ring attack the electrophilic nitrogen. One of the pi bonds breaks, and we put those electrons on the other nitrogen as a lone pair. We get an intermediate where we have the nitrogen attached to that second benzene ring, along with the hydrogen at the same carbon and a positive charge on the adjacent carbon, and then even a very weak base, perhaps the conjugate base of our acid, can remove the proton to restore aromaticity, and that then gives you the Azo compound, which, if you had previously modified those benzene rings with some other functional groups, you'd have a decent chance of them, of the molecule being colorful. All right. So we'll go over that chemistry again next time. Thank you for bearing with me for the stories. Good luck on your exam, and I will see you next week. Yeah. So, okay, speaking of the exam, it closes on Tuesday. The late fee kicks in at 2 o'clock, and the testing center is closed on President's Day. So you have today, tomorrow, and Tuesday. Yes? You want to come up and ask? What?
Monday instructional season.
Oh, Tuesday is a pretend Monday?
Yeah.
Okay.
Dr. Price,
Yeah.
super quick. So say you have you have two ortho paradirectors that are paired to each other. How do you determine which one directs it?
The one that's the strongest donor
Okay.
wins.
So like, is that just like the more electropositive atom?
Well, we talked about that, I think, at the beginning of last time.
Yeah.
Basically, nitrogen's a better donor
Yeah.
than oxygen
Yeah.
because of its lower electronegativity.
Yeah. Awesome. Thank you.
Yeah.
Thanks for
Hey.
the stories. That was really fun. Would you mind updating Learning Suite?
So, yes. Yeah, I'm so sorry. I haven't. The due date for the study guides and practice exams, or saying that you completed them, is usually on either the last day of the exam or later. I just haven't put them up yet, but no, don't worry about it. I'll email you when I've done it so that you can know.
Sounds good. My sanity is in part stabilized by the what-if calculator.
I see. Okay, I got you.
Thank you.
Yep, no problem.
Just a quick question. So the exam is just going to be this